Lebih main. Aduh, saya mantep ya. Good evening, people. Good evening. I think you can hear me clearly. Um, People, please take BioEve 12 paper. Take BioEve 12. Right, people, let's mark the first page answers. I want you to mark it. Can you mark first 7 answers, Lamai? Can you mark first 7? Okay, you're marking, right? Good. Right, just give me 1 minute, people. Right, first question. The the ability of organism change over time as a result of genetic modification, I'm taking as number 5. What is that, people? Evolution. Second one, ability to respond to stimuli received from internal and external environment. Second one is the irritability, correct, irritability. Internal and external environment, irritability. Which of the following physical properties enable water to act as transport medium. Water to act as transport medium. Water to act as transport medium. Water to act as a transport medium. Which of the following physical property enables water to act as a transport medium? Cohesion and addition. We'll take that. Ne? Third one is the answer. Correct. Question number 4. The property of water that prevents overheating by sunlight. High specific heat, high heat of vaporization, property of water that prevents overheating by sunlight. Cohesion, adhesion, expansion upon freezing. What do you think, people? Is it heat capacity, or should it be heat capacity, or should it be sir, did you hand out BioEve 12 paper at the physical class only? I'm an online student. You send it to the group today. Or else you can download it now. The property of water that prevents overheating by sunlight. I think people second is better. Now you think of your body overheating from sunlight, evaporation rather than high specific heat capacity, right? Overheating. Leaf is prevented from overheating by sunlight by the cooling process, right, people? Right. So I think the high specific heat capacity also you can take, but I think people more relevant is the majority answer, right? I can go with that because uh, I think it's better. It's better, right? Ability to moderate temperature due to high specific heat, water will function as a thermal buffer in living systems and adequate body aquatic bodies. Due to high heat of vaporization with the minimum loss can release much heat. This helps the organism cool their body surface. Right? Cool their body surface. Due to high specific heat, temperature, buffer, overheating. Prevent overheating by sunlight is the question. High specific heat, water will function as a thermal buffer during temperature fluctuations on Earth. High heat of vaporization, minimum loss helps organs to cool their body. Overheating. Now, heating and overheating, cooling, I think that's more relevant. So I'm taking second one as the answer. All right, buddies. Okay. Question number 5. Structural carbohydrate with aldose. Structural carbohydrate with aldose. Structural carbohydrate with aldose as its monomer. Aldose as its monomer. Glycogen, what is available in glycogen? Glucose, aldose, but it's not structural, so wrong. Hemicellulose, pentose, and hexosugars, neither. Wrong. Then starch is storage, no. Insulin, anyway wrong, ne? Cellulose you can take, structural. So answer is 5. Question number 6. Which of the following compound the monomer is a ketose? Ah, good. Fourth one, inulin, right? That's a ketose. Why? Fructose, ne? It is a fructose polymer. Okay, next one. Which of the following test can be carried out to identify solution? to identify if a solution contain lysine. What is the lysine? Lysine is amino acid, right? Lysine is amino acid. So it does not answer for Sudan 3 test. Sudan 3 test, lipids answer. Benedict test does not answer for Benedict. BioEve test does not answer because only proteins answer, not amino acid. Iodine test, starch can answer, right? So the answer is going to be none of the above. Fifth one, you all wrong. Huh? Right. Question number 8, people. If you have any arguments, send me. Sir, they have clearly given high heat of vaporization prevent overheating, right? Prevent overheating by the property of water. That's why I said now there are two statements. So what it says, water can absorb little high amount of heat by slight change in its own. Due to high specific heat, water will function as a thermal buffer and aquatic bodies during the temperature fluctuations on Earth. Due to high heat of vaporization, with the minimum loss of water of the an organism can release much heat. This is the help in cooling their body surface. Evaporation of sweat from the skin, maintain body temperature constant. Transpiration helps to cool the plant body and prevent becoming too warm in the sunlight. What is your argument? They have clearly given high heat of vaporization helps to prevent overheating. So what? So what? Property of water that prevent overheating the sunlight. So what is your argument? Then, so I'm taking second as the answer. Any issues with that? So what is lysine? Lysine is an amino acid. What is lysine? Lysine is an amino acid, right? So amino acid don't answer for BioEve test. BioEve test only the compounds with peptide bonds can answer. So when lysine, right? When lysine, didn't you learn lysine yet? No, eh? You didn't learn lysine, right? I think you're going to find this later. The amino acid solution contain lysine, leucine, methionine, cysteine, and all these are amino acids. I think it's better if I had given this as glycine. You learn glycine, right? Glycine you learn, eh? Lysine you're going to learn later. With the nutrition lesson, you find this lysine as an amino acid. Lysine, leucine, this is an essential amino acid, right? So amino acid can't answer for BioEve test. So none of the above should be the answer. Someone is asking, sir, why fifth answer for the seventh question? Fifth answer, none of the above, right? Vinuk. So what is your answer, Vinuk? What did you mark? What did you mark? So why BioEve test wrong? BioEve test, only polypeptide containing compounds answer for that. Do you see amino acids don't have peptide bonds? Amino acids don't have peptide bond, so they can't answer for that simply. Got it? So question, lysine contain protein. Which of the following can be carried out to identify a solution containing lysine? Big argument goes. Who's this? Right? To identify lysine, common test. Use so what is this? So what is your argument? More advanced and precise and less high. No, don't put these internet things on the chat line, right? I don't like that. That's useless. You argue with the resource book. Okay, so here, listen, lysine is an amino acid, right? What is answering for the BioEve test? What answers for BioEve test? Huh? Peptide bonds. So, do you see peptide bonds present in amino acid? Answer my question, right? Do you see peptide bonds present in amino acids, right? Dipeptide, when a dipeptide form only that it has amino acids. Sorry, the peptide bonds, right? When a dipeptide form, peptide bonds are there. So, dipeptide can answer for the BioEve test, but not a individual amino acid. Simple, that's all, right? Answer is 5. Okay, which is the, no, some of you are still arguing on the second one, properties of water that prevent overheating by sunlight. Now, I told you, right, high heat capacity can be helpful, right? The heat capacity is important to maintain constant body temperature without fluctuating the body temperature much. But then vaporization is going to be more helpful. So I'm going with the second one as the answer, right? Question number 8. Question number 8. Select the cell wall components of chlorophyta which is formed by aldose sugar polymerization. Select the cell wall component. So a solution of lysine means there will be a collection of amino acids joined by peptide bond. Ne, sir, no, not ne. It's not contain. Come on, it is just amino acid. Don't you know the difference between amino acid and a dipeptide? Right? Lysine is just simply what you call monomers. No peptide bonds in amino acids. Come on. This is amino acid. Hydrogen, NH2, R, COOH. You tell me. So where is peptide bond here? Can someone NH2, right? So can someone show the peptide bond here? Show me peptide bond. Come on, Mark can show peptide bond. Where is the peptide bond? So when two amino acids react, NH2, CO, CH, R, COO, OH, acid react with hydrogen, right? Then it's going to form, then it is going to form the double bond. Sorry, then it is going to form the peptide bond. Here, a peptide bond forms. So at least to have a peptide bond, it must be a dipeptide. Right? So dipeptide can answer for BioEve, that's correct. Right? What do you want? Do you want me to accept your answer and then give you marks, right? Without teaching the correct thing. It's like I have to now give just the marks, right? When you ask, I have to give you marks. Come on, take marks. You are correct. If it's wrong, I have to say wrong, right? If I am wrong, I'm not hesitating to, right, withdraw my answer. All right. So fifth one is the answer. Okay. Right. Then people, question number 7. Fifth one is the answer. Question number 8 to 11. Mark your answers now. Mark your answers, people. I like to see that you're arguing with me. I'm really happy about it. Not angry. Don't think that I'm angry with you. Right. Okay, thank you. So question number 8. Select the cell wall component. Select the cell wall component of chlorophyta, which is formed by an aldose sugar polymerization. Cell wall component of chlorophyta, hemicellulose, aldose sugar, pectin, inulin, peptidoglycan. It can't be peptidoglycan, right? Inulin is fructose polymer, ketose. Pectin, what is that? Galactronic, galactronic. Compound of chlorophyll which is formed by aldose sugar. So we'll take second one. I'll agree with your answer. Second one. Question number 9. Which of the following combination of polymers and its function is correct? Which of the following combination of polymer and its function is correct? Lactose. Polymer and its combination is correct. Lactose, storage. Combination of polymer and its function is correct. Lactose, storage. Polymer, it's not a polymer either. Storage is correct, but not a polymer. Inulin, transport, no. And then fat, respiratory substrate. Correct, but it's not a polymer, right, people? Fat is not a polymer, wrong. Insulin, blood glucose level homeostasis, correct. And insulin is a polymer, it's a polymer of amino acid. Glucagon, catalyst. No, glucagon is a hormone, not a catalyst. So fourth one is the answer. Fourth one is the answer. Very good. Which of the following statement is correct regarding the structure of DNA molecule? Which of the following statement is correct regarding the structure of DNA molecule? Structure of DNA molecules. Both strands of DNA molecules are similar. Both strands of a DNA molecule are similar. The nitrogenous base are paired on the outside of the helix. Both strands of the DNA molecule are similar, wrong, complementary, no. The nitrogen bases are paired in the outside to the center, wrong. And then the two strands are held together by hydrogen bonds between the nitrogenous bases. The backbone of the polynucleotide chain is composed of nitrogen bases and pentose sugar. No. One complete turn consists of 10 bases, the 10 base pairs. No. So we have the answer, third one. Very good. Question number 11. Select the correct statement. Surface features of a specimen can be observed by transmission electron microscope. No. You need scan for that. Using a scanning electron microscope, internal structure of the specimen, no. Practically, an electron microscope gives magnification of, what is this figure, 100 million, no. And transmission of electron microscope scatters many electrons from the specimen surface. No, that's done by the scan. Very thin specimen are used in transmission electron microscope. That's correct, absolutely correct. Fifth one is the answer. Good students. Very good students. Right? Good people, huh? Really good. Right? Beyond expectations, right? Right. So going to question number 13. Question number 13, my dear students. Right? My dear students. Question number 12 to 15. Who's going to mark? Who is going to mark? 15, 12 to 15. Can you give a small help? Okay. Harida. Okay, okay, okay. Question number 12. Sir, 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 proceed to the answer of a question. Also, proceed to what the chant in the voodoo. Hey, son, I can't understand your message. So there was a network issue for 11th, 10th and 11th. So the answers, please. Question number 10th and 11th. 10th we took third, right? The two strands are held together. They are asking correctly. Two strands are held together by hydrogen bonds between the nitrogen bases, correctly? Right? And then 11th we took 5th, ne? 11th, 5th. Right. So question number 11, third one is wrong. Nay, because it is paired nitrogenous bases. 11th, third, practically 11th. Third one is wrong because it paired nitrogen bases. Practically, an electron microscope gives magnification of, yeah, 11th or 10th, Muhammad, Arus. 10th, ne? Are you arguing about third one? Ne? Yeah, sorry, sir, for the 10th one. 10th one, third, two strands are held together by hydrogen bonds between the nitrogen bases. Two nitrogen bases, adenine and thymine, are held together by hydrogen bonds. Base pair or nitrogen bases. Two nitrogen bases, correctly? I don't see any error in that statement, right? Oh my God, look at the screen now. Select the incorrect statement about cancer. Cancer cells, uh, cancer cells will obtain growth factors required from the neighboring cells. That is wrong. Eh? We didn't say neighboring cells. Cancer cells have an abnormal cell cycle control system. Yeah. And then cancer cells do not consider the normal signals. Yes. Transformation process converts normal cell into an abnormal cell. Yes. Metastasis is the spread of the cancer cell to location distant from the original site. Correct me. Cancer cell. Correct, right? Fifth is also correct. They are asking incorrect one. So I'm taking first one as the answer for question number 12. Question number 13. Select the response. Select the response that correctly match the subcellular components and its function. Subcellular components and its function. Lysosome, detoxification, no. Smooth endoplasmic reticulum, metabolism of carbohydrates, correct. Golgi apparatus, synthesis of glycoprotein, Golgi glycoprotein, no. And then people, ribosome storage of calcium, no. Peroxisome converts sugar into fatty acid, convert sugar into fatty acid, no. So we have the answer. Second one, actually peroxisomes does a reverse, ne? Fatty acid into sugar. Sir, resource book says paired nitrogen bases. Doesn't matter, paired nitrogen bases, right? These are the nitrogen hydrogen bonds formed between nitrogenous bases. That is what it says, ne? I don't see that statement is becoming invalid due to absence of the word paired. Still, it gives the meaning. Okay, Muhammad Arus. And then we don't have any other alternative answer also. Right. Moving to next question. right? Arus, bring the book and come and talk to me, right? Um, Question number 14, select the correct responses regarding nucleic acid, right? Nucleic acid. Okay, sir, yeah, good. Select the correct responses regarding the nucleic acid. Nucleic acids are composed of backbone with repeating pattern of sugar. Nucleic acids are backbone of nucleic acid are composed of backbones with repeating pattern of sugar phosphate units. Right? I think I can agree with that. And they are linear polymers of nucleotides, correct? Linear, not branch, correct. Only thymine and cytosine are found in the nucleic acid as pyrimidine. Thymine and cytosine. Nucleic acid has uracil in RNA. So, 3 pyrimidines present: uracil, cytosine, thymine. Wrong statement. Always anti-parallel polynucleotide chains are found. Always anti-parallel polynucleotide chains are found. Always 2 anti-parallel. No, some of the nucleic acids can be single-stranded too, the RNA and all. They talk about nucleic acid, not DNA. Right? Nucleic acid can be either RNA or DNA. So, one of those two. Always the number of adenine bases is similar to the number of thymine bases in nucleic acid. Wrong. This can happen, people, in DNA, double-stranded DNA, right? So, otherwise wrong. So, we have the answer AB better. Question number 14, AB is the answer. Question number 15, which of the following statement regarding light microscope is correct? Cannot observe by naked eye. Cannot observe by naked eye. Regarding light microscope, cannot observe by. Which of the following statement regarding light microscope is correct? Cannot be observed by naked eye. I think, people, there is one word missing, neither. What is that? The specimen cannot be observed by naked eye. If that is there, specimen, at least that word, specimen. Then this is having some meaning. But anyway, this is wrong. Actual color of the object cannot be observed. Wrong. You can see that. Then, what is the third statement? Lesser resolution power than electron microscope. Yeah, correct. Heavy metals are used for staining. No. Strong magnets. No. Answer is 3. Third one. Right, good. Students, I'm moving to last 5. Moving to last 5 questions. 16 to 18. Last 3 questions. Can you mark the answers? (16 to 18). Which of the following is difference between prokaryotic and the eukaryotic cell? Subcellular components are not found, not surrounded by membrane prokaryotic. Eukaryotic, there are only subcellular components surrounded. First statement is wrong. Some are bounded, some are non-bounded. Both types of organelles are there. Microtubules are absent. Prokaryotic, correct. Microtubules are present. Eukaryotic cells, correct. Second is okay. Only 70 ribosomes present, only 80 ribosomes. Third one is wrong. DNA does not bind with protein. DNA does not bind. DNA bind with proteins. I think it's wrong. Both we can see that DNA attached to protein present, but this histone protein is a different question. Right? So these are wrong. All organisms fix nitrogen, prokaryotes, no. Now we have the answer. Answer is second one. Question number 17. Which of the following is correct combination regarding functions of subcellular structures, nucleolar synthesis of rRNA? Nucleolar synthesis of rRNA. SCR synthesis of glycoprotein. SCR synthesis of glycoprotein. Correct combination regarding function of the subcellular structures. Nucleolar synthesis of rRNA. Correct, Ned. SCR synthesis of glycoprotein. That I think we said for RER. Peroxisomes convert fatty acid into sugars. Peroxisomes are converting fatty acids into sugars. No, it's detoxification. Ne? Breakdown hydrogen peroxide. Wrong. Answer for 15th question, please. Third. Question number 4. Lysosomes. Sir, can you please explain the fourth answer for the DNA does not bind with protein, DNA bind with protein. No, prokaryotic, they have proteins, but no histone protein. That is what it says. Ne? That is what says. I think prokaryotic, what they say? DNA, they have given DNA circular, free in the sun. This region is DNA not associated with histone protein, particular type, histone. DNA linear and contain. Later in the resource book, Lamai, later in resource book. Now they say histone protein absent. Later in the resource book, we say archaea DNA present some species. Histone protein in archaea, archaebacteria, some species they have. Then bacteria, histone protein absent. But that doesn't mean protein completely absent. You can learn it later, right? It says histone protein absent. They have specified. Why fourth isn't the answer? DNA does not bind with protein? No, they have not mentioned that the DNA is not binding with protein. They say that DNA does not bind with histone protein, right? DNA present, DNA of bacteria contain, or DNA of prokaryotes contain protein, but it is not the type of protein called as histone protein. All right? Gayagi, is that clear for you? Right? So you have to remember that. They specifically say histone protein absent. Why they specifically say that? Think about it. Then peroxisomes convert fatty acid into sugars. No, wrong neither. Peroxisomes, they don't do that. Fatty acids into sugars. No, this is done by glycosomes. And then the lysosomes, detoxification of peroxides. No. And Golgi apparatus, exocytosis of residue materials. No, that's by lysosome. So we have the answer for 17th. First one. Question number 18. Select the correct statement about the cell organelles. Synthesis of phospholipid is done by RER. Synthesis of phospholipid done by RER. They are asking correct name. Phospholipids done by synthesis. They say only wrong. Only word is wrong. RER is correct, but only wrong. Synthesis of hemicellulose for plant cell wall is a function of Golgi apparatus. Hemicellulose, who's fixing hemicellulose for the cell? Cellulose and non-cellulose. Cellulose and non-cellulose cell wall materials. Such as, what they have given, such as hemicellulose. Ne? So I think the statement B okay. Ne? They are asking correct statements. Right, let me check this because I can't remember that. They say that manufacturing of cellulose and non-cellulose cell wall components such as pectina. I remembered as hemicellulose. So then wrong. Harida, so I'm wrong there, right? They say that synthesis of hemicellulose, no, what is this people? Pectin. Wrong. Basal body of flagella has 9 plus 0 microtubule arrangement. Basal body has 9 plus 0, correct. And then the peroxisomes is a single membrane bounded vesicle with oxidizing enzymes. Peroxisomes, correct. And then starch granules are peroxisomes are having oxidizing enzyme, right? Starch granules are found in stroma of chloroplast and matrix of mitochondria. Starch granules are found in stroma of chloroplast. No, matrix of mitochondria, you don't see that. So we have the answer now. Third one is the answer. Right, moving people to last, yeah, 19th question, 20th goes to the other side. Any doesn't matter. Can you answer 19th now, Lamai? Answer 19th. Select the correct statement regarding cell walls. So what about B? Synthesis of hemicellulose, that's wrong, ne? Resource book says pectin, so it's wrong. B wrong? C and D only. All right. Resource is pectin. Question number 19. Select the correct statement regarding cell walls. All prokaryotes have cell walls consisting peptidoglycan. All prokaryotes have cell walls consisting peptidoglycan. All prokaryotes have cell walls containing peptidoglycan. I think it's wrong. So, what are the organelles that synthesize phospholipids? What is the other one? I think it's given for RER and SER both. It's given for RER and SER both, right? Membrane factories and synthesis of, look at this, RER, synthesis of membrane phospholipids, SCR, synthesizing of lipid including oil, steroids and phospholipids, given for both, right? Then chemical composition, prokaryotes have cell walls consisting, all prokaryotes have cell walls consisting peptidoglycan, that is wrong, right? You're going to learn later for the later that the cell wall component of archaea and eukarya some difference. They say in your resource book under cell structure, peptidoglycan present in bacteria and cyanobacteria and polysaccharides and proteins present in archaea. So it's mentioned, right? Second one, chemical composition of the cell wall is similar in cell types of same species. Chemical composition of the cell walls is similar in cell types of same species. No, different cell types, composition is different. You can't agree with that. All protists possess cell walls mainly composed of cellulose. Wrong, neither some members. You haven't learned that part anyway yet. You're going to learn it soon. In plant cells, adjoining cell walls join through plasmodesmata. Plant cells, adjoining cell walls join through plasmodesmata. Adjoining cell walls join through plasmodesmata. Do you agree with that? Adjoining cell walls join through plasmodesmata. Cytoplasms, right? In plant cells adjoining cytoplasm. The secondary cell wall is deposited interior to the primary cell wall. This is correct. So I'm taking fifth one as the answer. Fifth is the answer. Secondary cell wall deposit interior to the primary cell wall is correct. You learn it, ne? In plant cell, adjoining cell wall join through plasmodesmata. Wrong, not cell wall, people. What is that? Cytoplasm. Right? Cytoplasms are going to connect it. Right. Question number 20. Question number 20. Question number 20. Can you mark? Lamai, can you mark that for me? All right. In homologous chromosome, pairs of binding of both sister chromatids and chromosomes, kinetochore microtubules, extending from the pole occur in the base. Pairs binding of both sister chromatids of a chromosome to the kinetochore microtubules. In homologous chromosomal pair, binding of both sister chromatids of a chromosome to kinetochore microtubules extending from one pole. Metaphase 1, Lamai, that's correct. You are very good. Very good students. Very good students. All right, my very good students. We are moving to the next structured part. Structured part. Name 3 areas of study considered under primary branch of biology. What is that? Botany. Botany. Zoology. Microbiology. Microbiology. Right. Second one, people indicate whether each of the following statement is correct or incorrect. Scientists assume that there are about 10 to over 100 million species. Correct. Most of the organisms begin their life as a single cell. Not most, Nedalamai. What we said for that? Is it most? Do you agree with this? Is it most or is it all? All. Wrong. Movement of organisms occur as a result of irritability and coordination. Yes. Reproduction is the ability to produce offspring for continuous existence of organisms. Is it continuous existence of organisms? Can one organism continue its life as a result of reproduction? No. Existence of a species. Species, wrong. What is the final answer of the 19th? What do you took as 19th final answer? I can't remember. What is 19th? Ah, 5, 5, 5. We said 5, ne? 5, why? Question number 3. When water is in liquid form, it's very fragile. State the chemical feature of water which contribute for this. Hydrogen bonds. Hydrogen bonds. So, for the first 10 to over 100 million, 10 to, scientists assume, first 10 million, about 10 to over 100, 10 million to over 100 million. So, what makes the difference? Now, you want them to say million there also. Okay. I think people 10 to over 100 million. This million applies here also, as I feel. Don't you feel that? Okay. Scientists assume that there are about 10 to over 100 million. That's a good argument anyway, right? I thought of 10 million to over 100 million, but then if you don't mention that 10 after million, that is a weakness, neither. 10 million to over 100 million. 10 to, when I say 10 to, we can simply think of, no, confused with the word over. 10 to over. But that's the statement, eh? 10 to over 100 billion species, ne? is given. It says understanding biological diversity. Scientists assume based on their studies there are about 10 million to over 100 million. 100 million species in the world. About 10 million. Yeah, I'll take it as wrong then, right? I'll take it as wrong because then there has to be 10 million mentioned here, not 10 to over 100, right? So I'll take it as wrong then, right? So does it have to be exact word like that? No, thing is, we must try to get the meaning of it. 10 to over 100 million, right? No, my question is, when 10 million word is missing, does it change the meaning or not? Does it change the meaning or not? 10 million to over 100 million and 10 to over 100 million. Does it change the meaning or not? Huh? Does it change the meaning or not? Difficult to decide, but anyway, well, I feel like that it has some influence. Okay, right? Resource book gives 10 million to over 100 million, 10 to over 100 million, 10 to 100 million, 10 to over. Right? I think it's wrong. Right? Okay. Then question number 3: when water is in liquid form, it is very fragile. But anyway, I didn't notice that too. Okay, 10 million. It's, I think some importantly, some value is there. When water is in liquid form and it's very fragile, state the chemical feature of water which contribute for this. Hydrogen bonds. Someone asked, why not polarity? So, but in the resource book, it says 10 to over 100 million. Is it? In my version, it is given as 10 million to over 100 million, my version. 2020-22. What is mentioned in your book? Huh? My book, it's mentioned 10 million to over 100 million. It's stated separately. Listen to these people. Scientists assume that there are about 10 million to over 100 million species. So in the old book, this is 10 to over. Yeah, okay.
Oh,
Yes. So it's confusing. Yeah. So we had to go with the new book. I think that's like some corrections they have done. Right? Okay, okay. Just keep in mind. So these two editions, it's slightly different way it's given. Right. So that's their weaknesses. Ne? Right. So that's the confusion. Ne? Right. Then question number 4. 4 major properties of water is important to maintain life on Earth. State the property important in the instances below. Small insects can walk. Cohesion. Transpiration in plants keep the plant body surface cool. What is that? Ability to moderate temperature. Ability moderate temperature. Right. And the next one. Organism in aquatic bodies can survive during the winter. Expansion upon freezing. Expansion upon freezing. Water function as thermal buffer in living systems and aquatic bodies during the temperature fluctuations on Earth. Ability to moderate temperature. Right, next one, people. State of which property of water does the solvent property based on? Polarity. Polarity. Through a structure of glycine. Alpha carbon has hydrogen on both sides. Carbonyl group. Nitrogen. Hydrogen. Why amino acid molecules are described as amphoteric? Why they are amphoteric? Both alkaline nature and carboxyl group present. Both alkaline nature. So someone's microphone is on. Please, can you turn off your microphones, people? Please check your microphones and then turn it off. Both the alkaline nature and carboxyl group. Carboxyl group present. Carboxyl group present. Group present. State how hydrogen bonds form the secondary protein structure. Right, what are you going to write, people? Secondary structure, intramolecular hydrogen bonds are going to form. Intramolecular hydrogen bonds. Intramolecular hydrogen bonds between oxygen atoms of carboxyl groups. Carboxyl groups and hydrogen atoms of amino groups. Groups of the same polypeptide chain backbone. Of the same polypeptide chain backbone. I exceeded my time limit. We'll try to finish it fast now. State 2, other functions of organelles that transport proteins produced by ribosomes. What are you going to write? Synthesis of glycoprotein. Then membrane phospholipids, synthesis of membrane phospho. What is this? RER, ne? Two other functions of the organelle. What is this? RER. State components of cytoskeleton which aid. Three components are there, ne? Microtubules, microfilaments, and what is that? Intermediate filaments. Intermediate filaments. Then state 2, functions of lignin found in secondary cell wall. Cement and anchor cellulose fibers. Cement and anchor cellulose fibers together providing hard and rigid matrix. Hard and rigid matrix, giving extra support. Right, then the next one, people, following diagram shows chromosomes in one stage of the nuclear division. Label A to E. What is A? Homologous chromosome pair. Homologous pair. Homologous chromosome pair, right? What is B? Thought of centromere. Centromere. C. Kinetochore. D. Synaptonemal. Synaptonemal complex. E. Cystochromatids. How about taking wait, people? I think this is non-cystochromatids, isn't it? Non-cystochromatids. I think so. Non-cyster. Non-cyster. Non-cystochromatids. Red and blue, right? Non-cysters, huh? Right. Then question number 2. What is crossing over non-cyster chromatids of paired homologous chromosome break, exchange, and rejoin? Non-cyster chromatids of paired homologous chromosomes break, exchange, and rejoin at corresponding points. What is significance of crossover result genetic variation? And then state the difference between chromatin and chromosome. What do you can say? Condensation of chromatin form chromosome. Condensation of chromatin forms chromosomes. And then the last one, people, state functions of nucleus. You have to write here: control cellular activities, synthesis of DNA, synthesis of rRNA, tRNA, store and transport genetic information, something like that. Essay question, right? They are asking similar to mitosis means you have to write meiosis 2 details, prophase 2, metaphase 2, anaphase 2, telophase 2, like that. You must write that meiosis 2 information. All right, people, my time is over, right? I have to take the other 2025 batch, the class. I'm leaving now. Thank you very much for your participation, right? And then I'll see you soon. Bye.